So, good day everyone. Praised be Jesus and Mary. I am Jeffrey A. Rara and with me is Joy Vasalio and Lester Magallon. We will be discussing about microbial diseases of the respiratory system. But before we go deeper on that, let me refresh your knowledge on the respiratory system. So we all know that the respiratory system is composed of the upper and the lower. So the upper respiratory system is consists of the nose, the pharynx or the throat, the middle ear or the eustachian tubes. While the lower respiratory system is consists of the larynx, the trachea, the bronchial tubes, the alveoli, and the pleura. So there are defenses on our respiratory system. On the upper respiratory system defense, we have this coarse hair which filters large particles from entering the respiratory tract. So makita nato ka ng mga buhok sa ato ang ilong, it filters large particles nga who wish to enter on our respiratory tract. While the ciliated mucous membrane of the nose and the throat traps airborne particles and removes them from the body, while the lymphoid tissue, the tonsils, and adenoids provides immunity to certain infections. For the lower respiratory tract defense, we have the ciliary escalator who maintains the lower respiratory system to be sterile because of its action. Those microorganisms hoping to infect the respiratory tract are caught in sticky mucus and move by the mucociliary escalator. So, there are the upper respiratory system diseases are mostly self-limiting. So when we say self-limiting, it is a clinical term. It is a term for clinical medicine which refers to any diseases whose natural history is resolved without treatment or no long. These are those diseases who has no long-term harmful effects on the person's health. So it is often caused by bacteria and viruses in combination. So those upper respiratory system diseases are caused by bacteria and viruses. So these are some upper respiratory diseases: the pharyngitis or the inflammation of the pharynx, the laryngitis or swelling or irritation or inflammation of the voice box or the larynx, the tonsillitis, which is an inflammation of the tonsils, and the sinusitis, which is the inflammation of the sinuses. So we have these agents of upper respiratory tract infections: the S. pneumonia for pharyngitis and tonsillitis, the S. pyogenes for pharyngitis and tonsillitis. We have C. diphtheria for diphtheria, B. pertussis for whooping cough, and the common cold, the rhinovirus for common cold. So those are the agents for your upper respiratory tract infections. And that ends my introduction for the respiratory system. And now we will proceed to the next topic for this discussion.
So, good morning. Praised be Jesus and Mary. I am Lester Magallon, and I am going to continue the discussion about the bacterial diseases of the upper respiratory system. So, as you can see, this is the anatomical structure of the upper respiratory system. The upper respiratory system consists of nose, the pharynx, the middle ear, and the eustachian tubes. So, these are spots where infection can happen, and these are the structures you have to familiarize with. And then next, the upper respiratory system defense. The coarse hairs in the nose filter large particles from air entering the respiratory tract. So let me have this in vernacular. So ang mga balibo daw sa atong sulod sa atong ilong is mo nang ga-filter sa mga hugaw o rabog na na atong ma-inhale na mosulod sa atong respiratory tract. And the ciliated mucous membranes of the nose and throat trap airborne particles and remove them from the body. And then katong mga airborne particles is gonna trap sa ciliated mucous membranes and gonna remove para dili mosulod sa atong body. And the lymphoid tissue, tonsils, and adenoids provide immunity to certain infections. The next is the upper respiratory system diseases. Most respiratory tract infections are self-limiting. Self-limiting means self-recovering. So for example, na kay viral illnesses in the upper respiratory system, it means makarecover ka without taking any form of medicine. Mo siyang meaning sa self-limiting or self-recovering. And then this is caused by bacteria and viruses in combination. So next, let's understand some of the microbial diseases of the upper respiratory system. So the first one is the pharyngitis, which is the inflammation of the mucous membranes of the throat. Hence, it is called the sore throat. So pharyngitis meaning the sore throat. And then the laryngitis, which is the inflammation of the larynx and the voice box. And when this happens, this can affect your ability to speak. And then the next one is the tonsillitis. Also is the inflammation of the tonsils. And tonsils are the lymphoid tissues that are found in the upper respiratory system. And then the next one is the sinusitis. Also the inflammation of the sinuses. And when this happens, heavy nasal discharge of mucus occur, and it can drain into the nasal cavity. And then the next one is the epiglottitis, the H influenza type B. So epiglottitis also is the inflammation of the epiglottis. An epiglottis is flat-like structure of cartilage that that prevents ingested materials from entering the larynx. So this epiglottitis, this is probably the most life-threatening infectious diseases of the upper respiratory system. Why? Because when this happens, it is rapidly developing diseases that can result in death. This is caused by opportunistic pathogen, which is pathogens usually Haemophilus influenzae type B. And then the next one is the streptococcal pharyngitis. So these are some of the bacterial diseases of the upper respiratory system. So kaganiya mo ni siya ang kanang something microbial diseases of the upper respiratory system. So kani karon is mo ni siya ang kanang the bacterial diseases of the upper respiratory system. So the first one is the streptococcal pharyngitis, also known as the strep throat. So meaning is an infection of the back of the throat, including the tonsils caused by a Streptococcus pyogenes. So their pathogenicity is enhanced by their resistance to phagocytosis, which can also able to produce special enzymes. And streptokinesis lies probein clots, and streptolysins are cytotoxic, which are toxic to the surrounding tissues or cells. Then, as you can see, here is a picture of a person having a streptococcal pharyngitis. Note the inflammation of the upper part of the mouth, which is basically right here. So as you can see, the readiness, this is called the pharyngitis or the sore throat. And then next, now when the streptococcus pyogenes causing streptococcal pharyngitis producing a reddening toxins, this resulting infection is called scarlet fever. So Also, scarlet fever is Streptococcus pyogenes, and also scarlet fever is pharyngitis or the sore throat. And the erythrogenic toxin produced by lysogenized Streptococcus pyogenes. So, lysogenized, the term lysogenized comes from the word lysogen, which is a type of life cycles that takes place where a bacteriophage infects certain types of bacteria. And then the next one is the diphtheria, caused by Corynebacterium diphtheria, a gram-positive rod diphtheria toxin produced by lysogenized Corynebacterium diphtheria. So here is the microscopic view of the Corynebacterium diphtheria. So the organisms can be pleomorphic. Pleomorphic means dili pare-parehas ilang something ilang size o ilang shape sa ilang cells into their nuclei. So the image shows the club cells. The club cells are observed often folding. So as you can see, more siya kanang something yung form is Fabi shape. And then the other one is observed. So makita ginato nga dili gid parehas ilang mga size, dili lang shape. So parehas lang ni. And the next is the diphtheria membrane, fibrin tissue bacterial cells. So this is the example of a person having a membrane on tonsils. And the next is diphtheria can be prevented by DTaP vaccine or the diphtheria toxoid. And then the continuous diphtheria infected skin wound leads to slow healing ulcers. So one example for this is kabantay man or something ka-experience na mo ka ng something gimatagtiki mo, and then magnuka man na siya, and then once nga mubuto na siya something na ay ka ng something bangag bitaw sa iyang samad mo na inana ay something itsura sa ka ng cutaneous diphtheria. So next is the otitis media, which means the infection of the middle ear. So this is caused by Streptococcus pneumonia. So the incidence is 35%, followed by the Haemophilus influenza, and followed by the Mozzarella catarrhalis, and followed by the Streptococcus pyogenes, and followed by the Streptococcus aureus. So the incidence of Streptococcus pneumonia reduced because of vaccine. And as you can see here, this picture is the otostrophic view of the auditory tube. As you can see, when the pressure builds up, you will have a bulging eardrum, which shows right here. Kanisha. And then this can be very painful, and this bulging happens because of the inflammation. Then next, let's move forward to the viral diseases of the upper respiratory system, which is the common cold. And then according to research, there are over 200 different viruses, and the most cold viruses is the rhinoviruses, which is the incidence 50%, followed by the coronaviruses, which is 15 to 20%. And we all know that this coronaviruses causes all people suffers, and many people are dying. And then next, the signs and symptoms of common cold. So these are sneezing, runny nose, congestion, sore throat, and malaise and cough. And then the pathogens and virulence factors. Enteroviruses or the rhinoviruses are the most common cause. Numerous other viruses cause colds. And then the pathogenesis, Cold viruses replicate in and then kill infected cells. And then next, so this is the structure of rhinoviruses, the most common cause of colds. Then next is the epidemiology of common cold. Rhinoviruses are highly infected, spread by coughing, sneezing, fomites, or person-to-person contact. Diagnosis, treatment, and prevention. Signs and symptoms are usually diagnostic. Fluconazole can reduce duration of symptoms. Hand antisepsis is important preventive measure. So, lastly, this table gives you the overall summary of what we have talked so far. So as you can see, this table is divided into the four categories, which is the disease. Under the disease is the bacterial diseases, and what are the pathogen involved. So here the pathogen involves the symptoms and the treatment.
We have learned already about the respiratory system and the bacterial diseases of the upper respiratory system. So we will proceed to the bacterial diseases of the lower respiratory system. As we have known that lower respiratory system consists of larynx, which is the voice box, the trachea, the bronchial tubes, the alveoli, and the pleura. So, bacteria, viruses, and fungi can cause bronchitis or bronchiolitis. These are the infections of the bronchi. So, if this bronchiolitis will become a severe complication with pulmonary alveoli became involved, it will become a pneumonia. These are the bacterial diseases of the lower respiratory system. So, first is the pertussis from the Bordetella pertussis, the tuberculosis with the pathogen of Mycobacterium tuberculosis, and the common bacterial pneumonias, which is the Streptococcus pneumoniae that causes a typical pneumonia, and the atypical pneumoniae, the Haemophilus influenzae, the Mycoplasma pneumoniae, the Legionella pneumophila, and Chlamydophila psittaci. So first, let us tackle about the pertussis or the whooping cough. This pertussis is caused by Bordetella pertussis, a gram-negative Coccobacillus bacteria, and is highly contagious. So the various toxins, the tracheal cytotoxins which damage the ciliated cells, and the pertussis toxins which enters blood to systematic symptoms. So there are three stages of this whooping cough. And the first stage is the catarrhal stage, which resembles a cold. Second is the paroxysmal stage, due to accumulation of mucus in trachea and bronchi. And proceed into the deep paroxysmal coughs, which brain and eye hemorrhage. And the last stage is the convalescent stage that can last for months. And laboratory diagnosis based on isolation of bacteria on enrichment and selective media followed by serological test. And the vaccination available, which is the DPT and new acellular DTaP. Next is the tuberculosis. So, tuberculosis has the pathogen have many pathogens. So this the Mycobacterium tuberculosis transmitted from human to human via aerosol. And one of the pathogen is the Mycobacterium bovis, which is more common in the cows and less than 1% in the US cases, usually extrapulmonary, affecting bones or lymphatic system, or known as the PAT disease. So the second pathogen is the Mycobacterium avium intracellular complex that infects people with late-stage HIV. And this mycobacteria is resistant to drying and disinfectants. So, the prevention is the BCG vaccine, which is a live, a virulent Mycobacterium bovis, which was extracted from the cow. So, the tuberculin test or tuberculin mantox test inject PPD and wait for delayed hypersensitivity reaction. But the problem is the BCG vaccination because when you conduct this test and if the person were injected with a BCG vaccine, the result of the test will become also a positive. So, not all positive in this tuberculin mantox test is really having a tuberculosis disease. So, the next slide will show you a video about the tuberculosis pathogenesis.
Tuberculosis, also known as TB, is spread through the air from one person to another. When someone with pulmonary TB disease coughs, TB bacteria are expelled into the air in tiny water droplets. These droplets can remain floating in the air for several hours, making it possible for someone nearby to inhale them. Once a person has breathed in droplets that contain TB bacteria, they travel down the trachea and enter the lungs, where they collect in the alveolar sacs. Once in the alveolar sacs, the bacteria begin to multiply. The body's immune system starts working and macrophages begin to surround the bacteria. A granuloma is formed to keep the bacteria from spreading. The TB bacteria remain in the lungs, but the body is protected from disease by the granuloma. In 8 to 10 weeks, the person will most likely test positive for latent TB infection. The progression from latent TB infection to TB disease occurs when the granuloma break open and the TB bacteria multiply. Then the person becomes sick with TB disease and may be infectious. This progression can occur immediately after infection, many years later, or not at all. When the TB escapes from the granuloma and begins destroying a person's lungs, it's called pulmonary TB. TB bacteria can also enter the bloodstream and travel to other parts of the body, causing extrapulmonary TB.
So that is the pathogenesis of the TB. As we have known that the prevention of tuberculosis or the TB is through the vaccination of the BCG or the Bacille calmette-guerin, which from the attenuated vaccine of Mycobacterium bovis, which is extracted from the cows. So this TB diagnosis by acid fast or FA of sputum. Then the chemotherapy of tuberculosis. So it takes 3 or 4 drugs taken for at least 6 months. But MDR-TB becoming prevalent. This MDR-TB means multi-drug resistant of tuberculosis becomes prevalent because some of the patients are not keen enough to follow this 6 months therapy. So, the solution is the DOTS. DOTS, which means the directly observed therapy short course. So, DOTS has 5 key components. So, the first is the political commitment. The second is the good quality diagnosis, the sputum smear microscopy. And third, the good quality drugs. There are 2 most powerful drugs for this tuberculosis, namely rifampin or rifampicin and isoniazid. So, fourth, 6 to 8 month therapy given under direct observation. So, the last component is the systematic monitoring and accountability to really stop the tuberculosis. So, tuberculosis in 2005. Each year, 1% of the global population is infected and 5 to 10% of infected get sick or infectious. So 1.6 million died in 2005 and populations affected is from Africa, which is 35%, Americas 18%, Europe 15%, Southeast Asia 44%. So the next disease is the melioidosis. Melioidosis is a bacterial disease which has the causative agent of the Burkholderia pseudomale. So the reservoir is the soil, especially the moist soil. So it is mainly in Southeast Asia and Northern Australia. And the diagnosis is through the bacterial culture and the treatment is ceftazidime. So this melioidosis is common in Southeast Asia and Northern Australia. The most frequently present as pneumonia, but also seen as tissue abscesses and severe sepsis. So the next disease is the typical pneumonia called the pneumococcal pneumonia. And this pneumococcal pneumonia is from the encapsulated pneumoniae and can be identified by production of alpha hemolysins, inhibition by optochin, bile solubility, and through serological tests. So, the mode of transmission is through the aerosol inhalation from asymptomatic carriers, which may cause illness due to immune suppression, smoking, viral infection. And the symptoms of pneumococcal pneumonia are the fever, breathing difficulty, chest pain, rust-colored sputum, and 80% of the bacterial pneumonia is the typical pneumonia called the pneumococcal pneumonia, especially in elderly. So the treatment is the penicillin, but multi-drug resistant is increasing and vaccine for 23 most common strains. Next is the mycoplasmal pneumonia that is also known as the primary atypical pneumonia or the walking pneumonia. So, Mycoplasma pneumoniae is the pathogen of Mycoplasmal pneumonia. It is a pleomorphic wall-less bacteria. So, Mycoplasma produce small fried egg colonies after 2 weeks incubation on enriched media containing horse serum and yeast extract. So, it is common in children and young adults and often mild enough to go undiagnosed for long periods of time. So, it is also called walking pneumonia because it means that walking pneumonia means that it is an informal term for pneumonia that isn't severe enough to require bed rest or hospitalization. So the diagnosis of mycoplasmal pneumonia is through the PCR or the polymerase chain reaction or serological test in the immunoglobulin M antibodies. So the next atypical pneumonia is the legionellosis or the Legionnaire's disease. The Legionella pneumophila is the pathogen of the legionellosis. It is a gram-rod bacteria and first discovered in 1976 among a group of elderly men attending an American Legion convention in Philadelphia. So the bacteria grow in water, such as pools, lakes, water systems of buildings, air conditioning units, and etc., then disseminated in the air. And the transmission is through inhalation, the aerosols. So there is no person-to-person transmission, and the diagnosis is through the bacterial culture and the acid fast test and the DNA probes. So the pneumonia and pleurisy is 15 to 20% mortality rate when hospitalized. So the treatment is erythromycin. So these are the viral diseases of the lower respiratory system. So these viral diseases, several viruses can cause pneumonia as complication of infection such as influenza, measles, or chickenpox. And etiologies are not usually identified in clinical laboratory because of the difficulty in isolating and identifying the viruses. So viral etiology suspected if no cause determined. So, the first viral disease is the RSV or the respiratory syncytial virus. It is most common cause of pneumonia in infants and 4,500 deaths annually. So, thus, it causes cell fusion. This cell fusion called syncytium. So, hence the name respiratory syncytial virus. So, causes cell fusion in cell culture and symptoms. The symptom is coughing and it is diagnosed by serological tests for viruses and antibodies. So, the treatment is ribavirin. If it's really life-threatening, the other treatment is the palivizumab. So the next viral disease is the influenza. So influenza virus, from the influenza virus, the ssRNA has 8 segments. So the symptoms of influenza are chills, fever, headache, muscle aches, and there are no intestinal symptoms. So the viral strains identified by antigenic differences in the H and N spikes. So also divided by antigenic differences in protein codes. So the type A, which mammals and birds were most severe and extensive. So currently, most common antigenic virus variants of influenza A virus are H1N1 and H3N2. So the second type is the type B and C, which for the humans only. So there are two types of influenza virus. The type A is the for mammals and birds. The type B and C is only for humans. So the viral isolates identified by HI and IF testing with monoclonal antibodies. So this is the H and the N, the hemagglutinin and the neuraminidase. So hemagglutinin, the H, spikes used for attachment to host cells, while the N or the neuraminidase, the spikes used to release virus from the cell. So as we can see that the green spike is the N spike, which is the spike used to release virus from the cell. And the H spike is the blue spike. And this spike, the hemagglutinin, is the spike used for attachment to host cells. So, H and N are virulence factors and antigen. So, the mutation in H and N leads to antigenic shifts, major changes only for type A, and antigenic drifts, minor changes for all types. So, the natural immunity and vaccination were obsolete. So the prevention and treatment. So widespread epidemics due to antigenic shifts or that causes pandemics. So the symptoms and diagnosis were the complications often due to bacterial secondary infections. So 50,000 to 70,000 deaths per year in the US, and also Guillain-Barré and Reyes syndrome. So vaccine produced in chicken embryos, flu shot, and nasal spray live. There are 4 antiviral drugs currently approved by FDA to treat acute uncomplicated influenza. So these are the 4 antiviral drugs: the oseltamivir phosphate or the Tamiflu, the branded name Tamiflu, the zanamivir or with the branded name Relenza, and the peramivir, the Rapivab, the baloxavir, Marboxil, the Drosophlosa. Next is the severe acute respiratory syndrome or the SARS. The symptoms and signs were high fever, shortness of breath, and difficulty of breathing. It is later developed to dry cough and pneumonia. So, the pathogen is caused by a coronavirus called SARS virus. And SARS virus spreads via respiratory droplets. So the diagnosis is based on signs and symptoms of SARS. So treatment is supportive. So let us tackle the next disease, which is the fungal diseases of the lower respiratory system. So, fungal spores are easily inhaled. They may germinate in the lower respiratory tract. The incidence of fungal diseases has been increasing in recent years. So, mycosis in the sections below can be treated with amphotericin B. These are the mycosis, the four mycosis, the coccidioidomycosis, the pneumocystis pneumonia, the histoplasmosis, the blastomycosis. So the first fungal disease of the lower respiratory system is the coccidioidomycosis, which is also known as the valley fever. So this is caused by the Dioides emitis. So the airborne transmission is the mode of transmission of this fungus. So most cases are subclinical and some get respiratory infection with flu-like symptoms. So in less than 1% of cases due to predisposing factors such as fatigue, poor nutrition, etc., is a progressive disseminated diseases from form resembling the tuberculosis. So, the diagnosis of this disease is through the serological test, and 97% of reported cases are from the California and Arizona. The second fungal disease of the lower respiratory system is the pneumocystis pneumonia or the PCP. So, it is from the pathogen or the fungus Pneumocystis cerevisiae or the P. carinii, a tiny fungus commonly found in nature, healthy human lungs and animals. So, it is transmitted through the aerosol inhalation and illness and death in newly infected infants and immunosuppressed individuals. So it is used to be leading cause of death in AIDS patients. So now it has a preventive drug therapy. So the diagnosis is through the detection of cysts in sputum samples. Next fungal disease is the histoplasmosis. This histoplasmosis, which is caused by Histoplasma capsulatum, widespread in Ohio and Mississippi river valleys, that infects body organs through bloodstream. So the risk factor is through the exposure to bird or bats droppings. So the diagnosis is through by isolation of fungus and treated with amphotericin B. Next fungal disease is the blastomycosis. This blastomycosis were caused by the blastomyces dermatitidis. So, this is a systematic yeast-like fungal infection commonly found in decaying wood and soil. Because this fungus thrive in the wet conditions, the symptoms of this disease are fever, loss of appetite, weight loss, eye discharge, and inflammation, difficulty of breathing, skin lesions filled with a pus. So it is a long infection that can occur after a person inhales the fungus. It is airborne. So fungal spores from the environment, however, many people who inhales the spores do not get sick. So it is treated with amphotericin B. So these are the microbial diseases of the lower respiratory system that we have learned in the previous slides. So the bacterial diseases are the pertussis or the whooping cough, the tuberculosis, the melioidosis, and including the pneumonia. So this pneumonia have two types, the typical pneumonia and the atypical pneumonia. So the typical pneumonia, which is also known as pneumococcal pneumonia, and the atypical pneumonia, we have tackled two, the Mycoplasma pneumoniae and legionellosis. The viral diseases of the lower respiratory system, which includes the respiratory syncytial virus or the RSV and the influenza. And the fungal diseases, which includes four, the histoplasmosis, the coccidioidomycosis, the pneumocystis, the pneumonia, and the blastomycosis. So this is the end for the chapter 24, which is the microbial diseases of the respiratory system.